The tyrosine phosphatase SHP2 is a key mediator of cellular signaling, promoting cell survival and proliferation. My team is focused on finding small molecule SHP2 inhibitors for the treatment of cancer.
Our technique provides a rapid and powerful method to establish cell penetrance as well as target engagement of small molecule SHP2 inhibitors in cells. This assures discovery resources are efficiently directed.
SHP2 is an attractive target for cancer therapy, and several SHP2 inhibitors are in clinical trials. Our technique will facilitate the discovery of next-generation inhibitors with improved efficacy profiles. Demonstrating the procedure will be Celeste Romero, a research assistant, and Douglas Sheffler, a research assistant professor from our laboratory.
To begin, detach HEC293 T cells from the plates using 3 milliliters of cell detachment reagent. Dilute the cells with 12 milliliters of growth media and collect them by centrifugation at 1,400 times G for 4 minutes. Resuspend the cell pellet in 10 milliliters of growth media and measure the concentration and viability of the cells with trypan blue and a cell counter. Plate 700,000 exponentially growing cells into each well of a 6-well culture plate and incubate them for 24 hours at 37 degrees Celsius and 5% carbon dioxide. On the next day, dilute 2 micrograms of plasmid DNA into 200